wrap up these next two lectures here. This lecture and the one that comes right after it will close out Unit 1, believe it or not. That will mark the end of the series of lectures for Exam 1. So this lecture and the other lecture you'll be watching this week close out the unit of Exam 1, and the material presented here and gone over in class will be reflected in Exam 1. So we're going over Chapter 4 today. The title of that chapter is Sex Determination and Sex-Linked Characteristics. In general, what we'll be covering in this lecture is how gender is determined in different animals. We'll give some specific examples and focus most on mammals, so different sex-determining systems, the gender-determining systems. Talk, as I said, a little bit in detail about how we determine gender in humans. From there, we'll move on to the genetics that are governed by sex chromosomes and sex-linked traits, how those genetics differ from the standard Mendelian genetics we've been discussing so far. And we'll close out with a discussion on Barr bodies. These are inactivated X chromosomes. We'll define them and talk about Mary Lyon, the scientist who hypothesized, and then it was proven that her hypothesis was correct, hypothesized the role of Barr bodies, what Barr bodies were. So it's actually the next three lectures, broken up by the exam after the second lecture in this series, that are used to discuss when Mendel's laws don't seem to be holding up as true. Now, I think you'll see, and it's somewhat fascinating to see, that Mendel's laws actually do hold up. What's going on is that chromosomes and other genetic elements aren't behaving in a way we expect them to. But once we understand the behavior of these chromosomes, we see how that they're being inherited to the next generation. We see that Mendel's laws, in fact, do explain all of the genetics we're observing. So the next two lectures after this one are based on Chapter 5 in our text, which is extensions and modifications of Mendel's basic principles. And as I said, I think you'll find that material very interesting. So in general, these exceptions are sometimes due to chromosomes not behaving in the way that we expect them to. But once we see how they behave, we understand how Mendel's laws hold true. As you know, we all have 22 pairs of chromosomes that we get from our parents. That's 44 chromosomes total, referred to as the autosomal chromosomes. But each of us have 46 chromosomes total, so there is a 23rd pair that we refer to as our sex chromosomes. These are the XX and XY chromosomes that I'm relatively certain that you're all familiar with. These chromosomes, the 23rd pair, determine our gender. However, more importantly, the 23rd chromosome pair does a whole lot more than just determine our gender. There are genes and traits encoded on the 23rd chromosome pair, and because males and females differ at the 23rd pair, and really that's the only set of chromosomes where males and females are significantly different, again, women are XX and men are XY, some really interesting genetics occurs at this 23rd chromosome pair, genetics that seems to defy Mendel's laws, but we'll see that they don't, in fact, because males and females have such different genetic information, such different genetic structures, chromosomes, at that pair. So that's what we're going to spend most of our time talking about today, the 23rd chromosome pair and how that impacts genetics. But before we get to that, it's important to just take a minute or two to discuss why are there genders and how can gender be determined? Most importantly, how is gender determined on all or at least different types of life on this planet? The textbook spends quite a bit of time at the beginning of this chapter going over some pretty specific details and pretty specific mechanisms that different organisms use to determine gender. And that's all very, very interesting. I wish I could cover it, but we just don't have time. So I'm going to summarize some of the most interesting examples for you here in the beginning of this lecture, but I encourage you, if you find this information to be interesting, go ahead and check out those portions of the textbook that I don't cover. You'll never be responsible for any of that on an exam, of course, but it might make for some interesting background information. In general, the word sex has two meanings, and it has two related meanings for very good reason. The first meaning of sex implies or defines that two organisms from a single species come together to contribute half of their genomes each to create a new whole genome constituting a new, novel, unique life form of that species. That's sex. That's making a baby through sex. It's a very dry definition of sex. It's the one that I prefer if ever I have to have this conversation with my own daughters, but this is, of course, the first meaning. Maybe the first meaning that comes to mind when we hear the word. But we use sex in a different way too. The second, although related, is different. It implies that the species doing this reproduction is divided into two different types. So here is where sex is a synonym for gender. Right? If you're filling out a form, it says, please indicate your sex, and the choices are male, female. So you're providing your gender for that particular form. In this species setup where there are two different types of individuals, only one of each type can reproduce. Only one of each type can donate DNA for reproduction. In other words, if we have type A and type B, we would call them male and female. Type A can't mate with type A. Type B can't mate with type B. You can only make a new individual when one type A mates with another type B. And again, we use terms males and females to indicate sex or gender. That's our convention. From a biological or genetic standpoint, the main distinguishing feature between males and females in all categories of life and all species is actually the size of the gametes. That's the distinguishing criterion. Males of any species tend to make the smaller gametes. This is even true of plants. And females make the larger gamete. That's how we define the gender. This is a true electron micrograph of a sperm about to fertilize an egg. The colors are false, but the image is real. And you can see the entire sperm, including most of its tail, is present here in this panel. And we only see, what do you think that is? Maybe 1/16 of the egg. The egg is that huge entire ball of cell. And so the difference in size between these two gametes is huge. Just you can see it right there. And keep in mind, we talked about this a little bit in a previous lecture when we talked about gametogenesis. Really all a male sperm is in this case is a capsule of half a genome. The tail, the sperm head itself, the entire structure is merely a delivery mechanism for genetic information. That's all that sperm contains that's of any value, is half of a human genome. The egg, on the other hand, is the cradle of this new life. It provides all of the cellular machinery, it provides nourishment, it provides the spark of life in that maternal cell. And that's why males and females have different gamete sizes. It's usually the female gamete, and then oftentimes the female organism, the maternal organism herself, that nourishes that new life and brings it to fruition. So we understand now, if we didn't before, that we need two types, two sexes, two genders in these types of species in order to get reproduction possible. The next question becomes, how do we get these two types? How are two types made? And that answer is different for different species. Different organisms do this different, but we always refer to the process the same way. Sex determination, the determining of sex or the determining of gender. Most sex determination is done by what we refer to as chromosomal sex determining systems. It's a lot of words, but the meaning I think is pretty obvious. This means that sex is determined chromosomally through the chromosomes, genetically. In 1891, some scientist, we'll leave the name out in this case, saw something weird, some weird structure within a single insect nucleus. Since it was weird and the scientist had no idea what the hell he was looking at, he called it the X-body, meaning the blank thing. I don't know what this is. I'll mark it with an X. Later, it was determined that this thing that was in the nucleus was a special sex-determining chromosome, and so it was referred to as the X chromosome, because now it was known that this was actually a genetic element. So the X remained, but we now call it a chromosome because that's what we know what it is. And that is the name of the X chromosome. That's the history of the name for the X chromosome. Many, many years later, another chromosome that was somehow involved in sex determination was discovered in nuclei, this time in mammalian nuclei. It was only seen in mammalian males, and it was given the next name in the alphabet, the name of a chromosome that comes after X, which was the Y chromosome. So oftentimes people think that the X chromosome here on the right is called the X chromosome because it's kind of shaped like an X, and the Y chromosome is called Y because it kind of looks like a Y. In this case, it would be the Y upside down here on the left. But that's not true. It's this history of calling the X chromosome the X body because they didn't know what else to call it. And what comes after X? Y. So we'll call the other one the Y chromosome. In any event, there are a number of different sex-determining systems that are chromosomally based in life. We're most familiar, of course, with the XXXY system. Almost every single mammal on this planet determines gender in this way, females being XX and males being XY. But there's also an XXXO system. Again, females are XX, but males are just X. The O in this case stands for 0. There's nothing else there. So in organisms that use this sex-determining system, females have 2 X chromosomes, males have 1 X chromosome, and that's it. Grasshoppers use this mechanism, as do other insects. We keep spreading on both sides of the alphabet because scientists really don't have any imagination at all, so we took the letter that came after Y, and we took the letter that became X, and defined another chromosomal sex-determining system that's primarily used in birds and reptiles, including snakes, also in butterflies. We call this the ZZZW system. What makes this system unique is that the pattern is flipped. In this case, it's the males who have 2 of the same chromosome, ZZ in this case, and the females have 2 different chromosomes. The females are ZW, but these are just placeholder names for chromosomes. ZZ is a chromosome that looks like any other. All of these systems are covered in some great detail in your text, if you'd like to go back and revisit it. We should also point out that in some species, not many, but in some species, it's more environmental cues that determine the gender. These cues determine if certain genders should arise, when those genders should arrive. Sometimes gender changes within the life of a single organism. An organism can flip from one gender to another. The question becomes for the species, what do you need now? Are you running low on females? Well, then let's bump up the number of females. Let's change so that we have more females. Are you running low on males? Maybe that's not as big a problem, but we can bump up the number of males too if we need to. The cues can be any environmental trigger. Temperature is a common one. Temperature affects gender determination in turtles and alligators. Other species members can trigger the flip in still other members of the same species. This is the slipper limpet story. These are the organisms that give rise to slipper shells that you might find at the beach. Any environmental cue could potentially affect gender determination. The slipper limpet story is somewhat interesting. We'll go over it a little bit briefly here, but just to give you a background on what that story is, this figure comes from your textbook directly. So a single larva of a slipper limpet lands on something that it wants to grab onto and live on. Could be a rock, could be a coral, something in the ocean, something in the water. And that larva settles on that and develops into a female. So pretty much in the absence of a cue, female is the default state. And that's going to be a reoccurring theme in this lecture. Female is the default state. That larva develops into a female. As I said, it releases chemicals, pheromones, hormones into the water that attracts other slipper limpets to the same spot. So now a second larval slipper limpet lands on top of the first female, but that second develops into a male. It develops into a male solely because it is on a female. If that second slipper limpet were to land on a bare rock, it would develop into female. That's the default state. But since it landed on a female, it develops into male. So those cues from that initial female trigger that new arriving slipper limpet to develop into male, and it stays male for a while. It develops into male long enough to mate with that original female, so that new slipper limpets can be formed. But then eventually, the signal from the initial slipper limpet fades, and that male reverts back to that default female state. So now you've got a second female on top of the first female. That second female releases her own signals, attracting new slipper limpets over to it that land on it, and now the process repeats. The new ones develop into male because of those signals being released, and they mate and make new slipper limpets, and then after a while those signals fade, and that new male reverts back into female. Now that it's female, it releases its own signals, attracting, so you get these kind of piling ons of female attracts a male, mating occurs, male then reverts to female, attracts a new male, mating occurs, that new male reverts to female, attracts a new male, and so you get this building up of generations on a single rock or a single coral. In any event, whatever the system is, the take-home message here is that sex, gender, is genetically determined. Actually, even in the case of the slipper limpet, the cues determined what the gender was going to be. But how that gender behaved, the secretion of the signals, the mating, and all of that, that's still encoded in the DNA. So all of the information for how to be the gender you are is in your DNA, and that's what we're going to take a look at today. When there are two types of genders, two sexes in a species, the type you are is either directly or indirectly due to your genes. So we're going to use flies, fruit flies, as a little bit of a deeper understanding. Fruit flies have only 8 chromosomes, 3 pairs or 6 autosomes, and then 1 pair of sex chromosomes, the same as we. Normally, fruit flies are like us. The females are XX and the males are XY, but something is strikingly different between us and fruit flies, at least in terms of gender determination. In mammals, it's the Y chromosome that makes you male. We're going to talk in detail about how that works and why that is in just a minute. But in flies, that's actually not true. The Y chromosome doesn't make a male fly male. In flies, the sex or gender determines more on the balance or the ratio of genes on the autosomes and the genes on the X chromosome. In flies, the X chromosome promotes female development, while the autosomes, those 6 other autosomes, promote maleness. It becomes a tug of war between the female promoting genes on the X chromosome and the male promoting genes on the autosome. And whoever has more numbers wins that tug of war. If the balance shifts in favor of the X chromosomes, you're female, because the X chromosome promotes female and they won the tug of war. If the balance shifts to the autosomes, then you're male, because the autosomes promote maleness, and if you have more of those, if they win that tug of war, you become male. Any balance of 1 to 1, meaning an equal number of X chromosomes and general autosomes. So you have 2 of each autosome pair, you have 2 of the X chromosome pair, that's a 1-to-1 ratio, then you're female. If it's a half ratio, in other words, you have 2 of every autosome pair, but 1 X chromosome, now you have twice as much autosome as X chromosome, or half as much X chromosome as autosome, then you're male. This chart breaks it down. What's interesting about the fruit fly story is that it's only the balance between autosomes and X chromosomes that matters. So we can see, looking at this chart initially, that XX is female. That's a 1-to-1 ratio. That's 2 of each autosome and 2 X chromosomes. Same number of each, you're female. XY is male, because now you have 1 X chromosome, 2 of each autosome. That's a half ratio. You have half as many X chromosomes as you do autosomes, so that's male. What's interesting about flies is if you take the Y chromosome away completely, and that's what the XO is referring to right here, you're still male. In the case of fruit flies, the Y chromosome is really just a placeholder, right? It's holding the place of the other half of the X chromosome pair. So if you take the Y chromosome away completely, experimentally, artificially, now the organism is X nothing, but still a totally fine functional male, because the ratio is still 1/2. 1 X chromosome, 2 autosomes, a 1/2 ratio. But you can get into really funky arrangements too. You can consider an XXY fruit fly. Well, we're only concerned with the ratio between the number of X chromosomes and the number of autosomes. Remember, the Y doesn't do anything. It's a placeholder. So if you're XXY, you have 2 X chromosomes, 2 of every autosome. That's a 1-to-1 ratio. You're female. If you add an extra autosome to every single autosome pair, you can screw with the ratio in funky ways that never naturally occurred. You can have an XX fly that has 3 of every autosome. Here, the ratio between X chromosomes and autosomes is 0.67. That's a non-naturally occurring ratio. There's no possible way this can happen in nature, but you can do it in a lab. And what you get is an intersex fly. You get a fly that shows some male characteristics and some female characteristics. So when you find the ratio right in the middle of 0.5 and 1, you get a fly that's right in the middle of male and female. So it truly is the balance. It does not, and I can't stress this enough, sex determination does not work this way in mammals. So the next question, of course, becomes, well then, how does sex determination work in us? In humans, it's the presence of the Y chromosome that determines maleness. Period. End of story. If a human being has a Y chromosome, they're male. If a human being does not have a Y chromosome, they are not male. Humans are very hormonal, and I don't mean that to sound funny. We are governed by our hormones. Much of what we are, who we are, what we do, how we behave, how we develop, our physiology inside our bodies, our physiology inside our cells, depends on the hormones that are circulating inside of us. And sex, gender determination, is no exception. Mammalian males are males because of testosterone, period. If you have testosterone, you're male. And if you don't have testosterone, you're not. You say, well, wait a minute now. I just contradicted myself. I just told you that it's the presence of the Y chromosome that makes you male, period. And now I'm telling you it's testosterone? Like, come on, pick one. I can't be both. If it's just one characteristic, it's just one characteristic. What's going on? Well, on that really small, attenuated, tiny, pathetic little Y chromosome is a very small gene that's called the SRY gene. The SRY gene, when it's expressed, sends a very strong signal to developing embryonic cells saying, make testes. That's what the SRY gene does. It promotes and sees to the development of testes in utero males, in male fetuses. Testes are made due to this signal from the SRY gene, again in the developing embryo. Testosterone is made by those testes. That testosterone is released into the fetal circulation system throughout the entire embryo. That testosterone, that hormone, causes the body to change, causes the brain to develop differently. That's the field of study, that's the passion of my wife's research, the other Dr. Manella in the basement of Carr. Cellular physiology has changed. The entire organism, from top to bottom, from inside to out, develops completely different due to the presence of testosterone. That testosterone came directly from the testes. The testes were made as a direct result of the SRY gene. The SRY gene was found on the Y chromosome. So the Y chromosome made that fetus male because the Y chromosome carried the SRY gene, which caused testes to form, which made and released testosterone, and it's the testosterone that made that individual male. So no Y chromosome, no testes. No testes, no testosterone. No testosterone, you ain't male. The default state of humanity is female. That's really profound to me. An unchallenged, unmanipulated human fetus that is allowed to develop the way that it would, without any outside interference, develops into female. We are all conceived female physiologically. We all begin our embryonic development as females. But some of us express SRY on Y chromosomes. Some of us then go down a male developmental pathway. This explains lots of different things that we see in physiology, including the age-old mind-bender question, why do men have nipples? Well, it's because that pre-programmed development begins to initiate before the testosterone is made. And we are all, male and female, and in some way, in some default way, female from our beginning. And I think that's pretty profound, pretty interesting. We can highlight this genetic determination of gender in humans by looking at a few different genetic syndromes in people, kind of drive these points home. I'm going to show you a few karyotypes. Karyotypes are the removal of human chromosomes right in the middle of mitosis when they're at their most compact and easiest to see. And then those chromosomes are arranged in their appropriate pairs. The biggest pair is the number 1 pair. The second biggest is the number 2, all the way down to the 23rd pair, the sex chromosomes. So it's just harvesting and lining up human chromosomes. So we're going to take a look at human chromosomes, and I'd like you to tell me what you think that these individuals are in terms of gender. The first is Turner syndrome. Let's take a quick look at this. Again, focus on the sex chromosomes because that's what we're focusing on now. You can see the autosomes, chromosome pair 1 through 22, look intact, healthy. They look the same in the 2 members of the homologous pair. And then we look at the sex chromosomes, the 23rd pair, and what do we see? We see 1 X chromosome and no Y. So what gender do you think this individual is going to be who has Turner syndrome? Keep in mind that the Y chromosome makes you male. You have the Y, you have SRY. You have the SRY gene, you make testosterone. You make testosterone, you're male. No Y chromosome, no SRY gene, no testosterone, no maleness. So this individual would develop as a female. Individuals with Turner syndrome have normal intelligence, no mental deficit. They're usually sterile. That's interesting. But they are usually sterile, unable to conceive. Oftentimes they have underdeveloped secondary sexual characteristics, so their breasts don't develop as much, their hips don't widen as much. So there are some physical features that are recognizable in Turner syndrome. You need an X chromosome. Women are supposed to have 2, so we see some straying away from what we would consider normal development. But for all intents and purposes, for all physiological measurings and all appearances, Turner syndrome individuals are female. Klinefelter syndrome. So again, we see normal healthy chromosomes from pair 1 through 22, and then we see triploidy at the sex chromosomes. That's 3 sex chromosomes, and this individual is XXY. What do you think the gender, the perceived gender of this individual would be? Well, keeping in mind that Y makes you male, this individual has a Y chromosome, so they're going to have the SRY gene, they're going to make testes, they're going to make testosterone, they will develop as a male. There is 2 X chromosomes, which are traditionally expected in a female, so we do see some diminished secondary sexual characteristics in these males as well. The testes are smaller than average. There's reduced facial hair. We see tallness. Usually there's sterility again. Usually normal intelligence. Another syndrome, triplo X syndrome. The name of the syndrome defines its genetics. There's 3 X chromosomes. So hopefully you realize that this individual would be female. For a very interesting reason that we'll end the lecture with, these individuals are often undiagnosed. They are so normal, quote unquote, whatever the hell that means. I don't know what normal is. I don't think there is anybody who's normal, but their appearance, their physiology, their mental abilities, they are extremely average. And so oftentimes people are triplo X and live their whole life without ever knowing that, because there's really no symptoms, no recognizable features of triplo X syndrome at all. But of course they are female. They don't have a Y chromosome, and so they do not have SRY. They don't make testosterone. They're female. So what does this tell us? Well, we realize a few things. Both sexes get and need an X chromosome. You need at least 1 X chromosome to be a viable human being. There has never been a human being that survived, that lived, that did not contain at least 1 X chromosome. So 1 X chromosome is critical for human survival. This strongly suggests that cells missing X chromosomes don't survive. Otherwise, by now, you think we would have come across somebody who had that condition. Again, to drive the point home, the Y chromosome makes you male, period. And in fact, it's the SRY gene that makes you male. You could, and it's been done in other mammals, transfer the SRY gene off of the Y chromosome onto some other chromosome, and the SRY gene makes that individual animal male. So if you have SRY, you make testes. If you make testes, you make testosterone. If you make testosterone, you develop into a male, period. It's just that the natural home of the SRY gene is on the Y chromosome. So naturally, Y chromosomes make you male. Female is the default state. In the absence of interference during development, in the absence of hormonal interference, human beings develop into female. Genes for fertility must be found on both the X and Y chromosomes, because individuals that are missing some of those, either missing a single X as a female, or missing their X chromosome missing the Y chromosome as well, are infertile, are sterile. And so we need the right balance of chromosomes in each gender to be fertile. For a female to be fertile, she needs 2 X chromosomes. For a male to be fertile, he needs 1 X and 1 Y. And we see that balance is important. Triplo X is often undiagnosed, but no healthy, quote-unquote, normal individuals are present in our population with more than 3 X chromosomes. So it is possible to have too much of a good thing. Too many X chromosomes does lead to significant syndromes, significant health defects. So I'm kind of sending 2 mixed messages at the same time, and I wouldn't blame you if you're trying hard to wrap your mind around it. I keep telling you that the Y chromosome makes you male. No Y no maleness, and this SRY gene plays a role in that. And then I keep coming back to these hormones too and talking about testosterone. So let's use a specific, interesting condition to kind of drive the point home of what the relationship is between hormones and genetics, hormones in the Y chromosome. So here's an individual on the right here. This is a medical picture, and that's why her face is blacked out. Her identity is hidden. What gender is this person? If you saw this person on the street, what gender would you identify them as? And I think it's obvious. This person's a woman. This is a female. This individual possesses a syndrome called androgen insensitivity, diagnosed as androgen insensitive. What that means is that her body cannot sense or respond to the hormone testosterone. We say, well, she's a woman. She doesn't have testosterone. Why should that be an issue at all, whether she can sense it or not? Well, I would hesitate to go down that road and say this is a woman. See, this condition leaves her with a physiology that is irresponsive to testosterone. It's almost as though the cells of her body are individual cell phones that can no longer pick up the signal from the cell phone tower. Calls can be made. Calls are being sent to the cell phones. Testosterone in this individual is actually being made. That testosterone signal is being sent to all of her cells. But those cells are broken and can't receive that testosterone message. This individual is an XY individual. Her cells are genetically male. She was conceived as a male. Her father gave a Y chromosome during her conception, pigeonholing her to a male development. But she had a separate mutation they're called receptors, in the proteins that are responsible for sensing testosterone. Those proteins are mutated. And so although she has testes, and although those testes are making testosterone, and although her blood is riddled with testosterone levels that are equal to the testosterone levels in a male, the rest of her body could never sense that testosterone. Therefore, those cells couldn't respond to the testosterone and go down that male developmental pathway. So she defaulted to the female state, because that's what happens to a human being that is left unchanged during development. Without the effects of testosterone being sensed and acted upon, without those effects being possible, this individual went down that default female developmental program. So I ask you again, now with all that background information, is this person male? It's a pretty thought-provoking question. It's actually the bridge between biology and society. So we can put on our biologist jacket and say, yes, of course, this individual is male. She's XY. Y chromosomes make you male. Genetically, this individual is male. But then as a human being, how dare we? This individual, no doubt, identifies herself as female, has felt female her entire life, has behaved female her entire life. There's a sense, a self-perception that is female. How dare we label someone as something else solely on the basis of their genetics? So these are the types of things we have to consider that go beyond the conceptual topics of the course, that go beyond the field of genetics. We're talking about people, and people don't always fit into these nice little boxes that we've constructed with science. It's just something to keep in mind. All right, so let's move on to the genetics of all this. Let's talk about some sex-linked traits. There are genes with alleles encoding traits that occur on X chromosomes. And we're going to use the example of white eyes in Drosophila as our first example. So everything we just talked about is great. It's all fine and good. But when we get down to the genetics of it, it pretty much gets in the way of everything we've learned about so far. We're going to see that the nice Mendelian genetics we've been talking about thus far in the course seem to break down when we consider genes that are encoded on the X chromosome. So far before this lecture, everyone we've talked about, whether it be plant, mouse, fruit fly, or a person, has been diploid. 2 alleles for each gene. One from mom, one from dad. Dominant, recessive, codominant, co-penetrant, etc., but 2 alleles for each gene. And all of that still holds true for you guys, for all females. Females have 2 X chromosomes, 1 from mom, 1 from dad, 2 of each allele. Some are dominant, some are recessive. All those rules apply. But what about me? Where do I fall into this? Well, males are haploid. Males have half that genetic information. Males are haploid for all of the genes that are found on the X chromosome because we only have 1. We have 1 X chromosome from our mother and the Y chromosome from our father, which is really just a delivery receptacle for an SRY gene. The genes that are found on the X chromosome are called X-linked. Those genes are linked or part of the X chromosome. And if a male has a mutant, non-functional allele on his one and only X chromosome, he's just SOL. Sorry, pal. Nothing we can do about that. Females have 2 options for X-linked traits because they have 2 X chromosomes. Remember that example of brown hair color in the factory we used quite a while back? Well, we said everybody has 2 machines for making their trait. One machine can be broken. The other machine most likely will still work. To lose the trait, you had to have 2 broken machines. And for all women, that's still true even for X-linked traits because women have 2 X chromosomes. But males only have 1 X chromosome. So males only have 1 shot. If males pick up a single mutant allele on their 1 X chromosome, that's the mutant trait they show. There's no other allele present on any other chromosome that can compensate for that. A long time ago, a pioneering geneticist, perhaps the most influential geneticist after Mendel himself, named Thomas Hunt Morgan, we've talked about him before, noticed a male fruit fly in his collection that had white eyes instead of red. Most fruit flies have red eyes. This particular mutant, who was male, had white eyes. From that observation on, the rest was history, and Thomas Hunt Morgan began to open our eyes to traits that didn't behave in the ways that Mendel predicted. What Morgan did was he took his white-eyed fly, his male fly, and he mated it with a true-breeding, homozygote, wild-type, red-eyed female. And in that F1, all the progeny were red-eyed. Okay, no big surprise. We've seen that before. That suggests that the male fly is homozygous recessive for white eyes, the female fly is homozygous dominant for red eyes, and you did the mating and everybody's heterozygotic. Okay, no surprise there. And then Morgan did what any good geneticist would do, and he went to the F2. He took the F1 siblings and mated them, and crossed that progeny of the F1, and did the next cross. The ratios he expected in the F2 are the exact same ratios we would expect. This is the cross that Morgan thought he was doing. This is the heterozygotic by heterozygotic F1 cross. And so 3/4 of those flies should have red eyes and 1/4 should have white. What Morgan saw was a gender difference. The males and the females showed different ratios. All of the females in the F2 had red eyes. Well, that's not expected. We expect 3/4 and 1/4. So all the females had red eyes. For the males, it was 50-50. Well, that's not what we expect either. For males, half of the males had the red eyes again, and half of the males had white eyes. This threw Morgan for a loop. Doesn't fit Mendelian predictions. So what in the world's going on? Morgan, in his astute insight, realized that to explain these ratios, the eye color gene must be on the sex chromosome, on the X chromosome. And here's why. Here's what Morgan proposed. That if we assume that the eye color gene is on the X chromosome, then my true breeding red-eyed females, right here, are wild-type, homozygote, wild-type, homozygote dominant. My white-eyed males, however, are haploid at the X chromosome because they're XY. And so they have 1 X chromosome carrying the white-eyed allele, which is recessive, and the other sex chromosome is Y, which doesn't do anything. So the reason why these males have white eyes is because they only have 1 allele for eye color, and that allele happens to be recessive. So let's imagine that cross. Now these are the sex chromosomes. So when we do that cross, all the daughters of this cross, all the females, are going to get 1 X from mom and 1 X from dad. That's what makes them female. The X they get from mom is going to definitely have the dominant red-eyed allele, because that's all mom had to offer. The X chromosome from dad carries the recessive white-eyed allele. That's all dad has to offer. But all the daughters will be heterozygotic. The X from mom has the dominant red-eyed allele. The X from dad has the recessive white-eyed allele. So all the females are going to be heterozygotic. Okay. For the males, to make a male, mom has to give an X chromosome and dad has to give the Y chromosome. All mom has to give for the X chromosome is the dominant red-eyed allele. And all dad has to give for the Y chromosome is his Y chromosome, which carries no eye color allele at all. So all the males have red eyes as well, because they got the dominant red-eyed allele from mom and the Y chromosome from dad. So there's your 100% red eyes in F1. Now we're going to do the mating of the F1 to make F2. All of these heterozygotic females are going to mate with all of these males. Well, the heterozygotic females can now either give the dominant red-eyed allele chromosome or the recessive white-eyed allele chromosome. These males can either give the X chromosome that has the red-eyed allele or the Y chromosome that has nothing. So let's carry it through for the daughters first. For the daughters, the only way we can have a daughter is if each parent gives an X chromosome. So the mom gives the dominant red-eyed allele X chromosome, the dad gives the dominant red-eyed allele X chromosome, and those daughters have red eyes, they are homozygote dominant. Or the mom gives the white-eyed allele X chromosome, but in order for this to be a daughter, the father gave his dominant X chromosome. So these individuals also have red eyes, but they are heterozygotic. So the daughters all have red eyes. Half of them are homozygotic, half of them are heterozygotic, but they all have red eyes. To make a son, the father gives his Y chromosome, which contributes no eye color information at all. Half the sons get the dominant red-eyed X chromosome from mom, and so they have red eyes. The other half of the sons that got the Y chromosome from dad get the recessive white-eyed allele from the mother, making them XY for gender, male, but making them white-eyed for eye color because they have the recessive allele from their mother and no information from their father. So 50% of your males are white-eyed and 50% are red. Morgan also did his cross, testing a white-eyed female with a red-eyed male. I'd like you to take a moment, pause the lecture if you need to, and tell me what he saw when he did this cross. Well, the only way a female is going to have white eyes is if she's homozygote recessive. She has two X chromosomes. Each X chromosome must have the recessive allele. That's what gives her white eyes. The only way the male shows the red-eyed trait is if his one X chromosome, the only one he's got, has the red-eyed allele. And now we're doing that cross. So we cross the homozygote recessive females with the heterozygotic males, and what we see is that every single female has red eyes. Because, and stay with me here, to make that female, the father had to have given his X chromosome. To make that female, the father had to have given his X chromosome. In this case, it doesn't matter which X chromosome the mother gave because they're the same. They both carry the recessive allele. But that makes every single daughter heterozygotic, showing the dominant trait, which is red eyes. But to make the son, the father, to make that son, had to, by definition, give his Y chromosome, which contributes no genetic information at all. The mother, again, gives the only X chromosome she can. It doesn't matter which one she gives, it carries the recessive allele. And the sons are faced with the recessive allele from mom and nothing from dad. That's what made them sons. And so all the males have white eyes. You can carry this to the F2. Now we have white-eyed males mating with heterozygotic females. Of the daughters, 50% of them will get one of the dominant alleles from the mother and one of the dominant alleles from the mother and the only recessive X chromosome allele from the father, giving them red eyes but making them heterozygotic. The other half will get the other X chromosome from mom, which carries the recessive white-eyed allele, and the only X chromosome dad has to give, which also carries the white-eyed allele, making them homozygotic recessive and white eyes. Where are the daughters that got the Y chromosome? There ain't none. If a kid got the Y chromosome, he's a son. So to have the daughters, the father only could give the X chromosome. That's what made the daughters. On the son side, however, to have the son, the father always gave the Y chromosome. No genetic information at all. For sons, the X-linked traits are dictated by the X chromosome the mother gives only. So half of those sons got the mother's dominant allele, giving them red eyes, and the other half of the sons got the mother's recessive alleles, giving them white eyes. And so we also see a 50-50 split in the sons. These are not our typical predicted Mendelian genetic ratios. These depart from what we would predict given Mendelian genetics. But as I told you in the intro, once we understand where these genes are, how those particular chromosomes are inherited, and what those chromosomes do, not only in terms of the trait, but also in terms of gender, it all makes sense. None of these observations prove Mendel wrong. In fact, they refortify what Mendel discovered. It's just that males and females are different at these chromosomes, so males show the one allele they have, whereas females continue to have the dominant and recessive relationship between alleles that we saw at other chromosomes. It all makes sense, right? I hope. If it doesn't, back the lecture up 5 minutes or so, try it again, and if it doesn't even after that, you got to put it in the reflection, got to bring it to my attention, you got to come to office hours, you have to seek my help. Got to get this to make sense for you, because we are rapidly building on the basic premise of Mendelian genetics, as you can see here, to more complicated things. So one way or another, let's work together to get this to make sense for you. So let's come to something a little bit closer to home. We obviously don't have red eyes and white eyes. Let's talk about some X-linked traits that are important to us that we find in humans. Colorblindness in humans is an X-linked trait. The gene that allows human beings, human eyes, to see red and green is found on the X chromosome. When this gene is mutated, when human beings have no functional gene for that particular protein, for that gene, that individual is red-green colorblind. This is what the intact rainbow spectrum looks like to someone with intact vision. I can see this fine. I'm not colorblind. These are the colors of the rainbow, Roy G. Biv. This very same panel looks like this to an individual who suffers from red-green colorblindness. They can't see the red and the green. It's all different shades of yellow. And so it's a pretty significant syndrome. It's a pretty significant effect, especially when you start to get to information containing things. This on the left is what a map would look like to somebody with normal vision. It probably looks this way to everyone that's watching this right now. On the right, though, is how the very same image would look like to someone with red-green color blindness. And you can see a lot of the information, a lot of the differences in these country color codes are lost when you have red-green color blindness. So, like most mutant alleles, the allele that causes this condition is recessive, but it's on the X chromosome. You only need one normal, functional, wild-type allele for this gene to have normal, intact vision. But because it's on the X chromosome, females have two shots at getting a normal allele. One from mom's X chromosome, one from dad's. That's what made them female, is that they got two X chromosomes. So on each of those two X chromosomes is this gene for color blindness, and so females have two chances to get a good allele. If they get one bad allele, no big deal, because they could get a good allele on the other chromosome. Males don't have that luxury. For this trait, males have one shot and one shot only. It's the X chromosome they got from mom. Whatever allele that one X chromosome has on it is the trait those males will express, because what they got from their fathers is a puny Y chromosome that has no gene for color blindness on it at all. So if mom gives her son a defective, mutated allele, he's colorblind. That's it. He's got no other chance. No other shot at another allele to balance that mutation. That's the end of the story. And again, why is it the mother's fault, quote unquote? Not that it's anybody's fault, but why is it the mother's genetics that's responsible for this and not the father's? You say, well, wait a minute, Dr. M. The kid's a male because of the father. The father dictated the gender. The father gave the Y chromosome. The father made him male. So you're telling me he's colorblind because he's male. The father made him male, but the colorblindness is the mother's doing? Well, yeah. Because the color blindness is on the X chromosome, and mom gave him that X. It's not what dad did to make the kid colorblind. It's what dad didn't do. Dad didn't give another X chromosome, which could have balanced that mutation. Dad instead gave an informationless Y chromosome, establishing not only the son's gender as male, but the son's lack of a second allele for colorblindness. So let's go over this again and make sure we all get it. I've got a few crosses here. So the first cross we'll consider on the left is a normal female mating with a colorblind male. And when we say normal for female, in this case, we mean true breeding. So she's homozygotic dominant. So she has two dominant non-colorblind X chromosomes to give, and the father is colorblind. The father has a mutant allele on his X chromosome. It's the only X chromosome he's got. His other chromosome, of course, is Y. That's what makes him male. So a pretty straightforward cross here. We have all of the children are getting the same chromosome from the mother. It's an X chromosome with the dominant non-colorblind allele. It doesn't really matter which one she gives in this case because both have the same allele. She's homozygotic. For all the daughters of this man are going to be heterozygotic. They have a normal color vision allele from mom. They have the mutant color vision allele from dad. So they carry that allele, but they show the dominant trait. They have normal vision. We would refer to these women as carriers for the trait, meaning they have the allele, but they're not showing the trait themselves. All of the sons, excuse me, are sons because dad gave them a Y chromosome. So they got the Y chromosome from dad, but remember, all mom had to give, all she had to offer, was a normal, non-mutated color vision allele. And so all the sons have normal color vision as well in this case. Let's do the same cross, but let's do it in reverse. The reciprocal cross. Now it's the mom who's colorblind, and it's the father who has normal color vision. Well, for males, that's easy. If the male has normal color vision, the male has a normal X trait, a normal allele on the X chromosome. Males always show the one allele they have. So the male has a dominant X chromosome, a dominant allele on their X chromosome, and an empty Y. The females, to show color blindness, have to be homozygotic recessive. Females are diploid at their X chromosome, and the only way they're going to show the mutant trait is if they have two mutant alleles, meaning they're homozygotic recessive for color blindness. Now let's do this cross. Same idea, the female only has one particular type of X chromosome to give. It really doesn't matter which one she gives. They're both mutant, so that's the one she gives. All of her daughters get one mutant allele from mom, because that's all she had to offer, and get the one X chromosome that dad has to give. That X chromosome happens to have the dominant trait on it. They are daughters because they got this X chromosome from dad, and they show normal color vision because they are, once again, heterozygotic. But on the male side, the father gives the Y chromosome to all of his sons. It is informationless in terms of this trait. The mother gives the only type of X chromosome she has to give, which is mutant, and so her sons, and this is important now, every single one of her sons will show the mutant trait. Because those sons got her one X chromosome, which was mutant, and that's the only X chromosome they got because they are sons. So a color-blind female with red-green color blindness is guaranteed to have nothing but colorblind sons. To me, that's pretty amazing that we can do that kind of conclusive definition with an organism as complicated as human beings. But I can guarantee you right now that any woman out there that has red-green color blindness, if she has and shows the red-green colorblind trait, every single one of her sons will be colorblind. Period. That's what the genetics says. Here's a sample problem we can work through pretty quickly together right here. In this question, Betty's female. Betty has normal vision, but her mother is colorblind. Bill is colorblind. Bill is Betty's man. Bill and Betty marry, they have a child together. What's the probability that the child will be colorblind? Well, we can take this story and break it down. We know that Betty is heterozygotic. Betty has normal color vision, so we know she's got a normal allele on one X chromosome. If she was homozygotic recessive, she'd be colorblind. So she has normal vision. What we don't know is, does Betty have two normal alleles? Is she homozygotic dominant, or is she heterozygotic? So then we keep reading. But Betty's mother is colorblind. Well, if the mother is colorblind, that means both of the mother's X chromosomes were mutant. Same as what we said here. The only way a female shows the colorblind trait, the only way the female shows any recessive X-linked trait, is if she's homozygotic recessive. So Betty's mom is colorblind. To make Betty, the mother had to give a colorblind allele. So Betty's carrying a colorblind allele from her mother, but it must be a normal allele from her father because she has normal vision. So Betty is heterozygotic. Bill is male, so we know Bill is XY, and Bill is colorblind, so we know Bill's one and only X chromosome is mutant. So Betty's a carrier, and Bill is carrying a recessive trait on his X chromosome. These are the gametes that Betty and Bill can make. Betty can donate an X chromosome that's normal, because she has one of those. Betty can donate an X chromosome that's mutant, because she got one of those from her mother. Bill can donate an X chromosome that's mutant, because that's his only X chromosome to give, and Bill can donate a Y chromosome as well for all of his sons. In this case, if we do the cross, we see that half of Betty and Bill's daughters will be colorblind. Half of them will receive a dominant X chromosome from mom, and the other half will receive a recessive X chromosome from mom. All of those children, if they're daughters, will receive dad's only X chromosome, which is mutant. So for these individuals, they will be normal in terms of color vision, although carriers and heterozygotic. These individuals will be colorblind because they got dad's only recessive chromosome for colorblindness, and they got mom's recessive X chromosome as well. Same ratios in this case hold true for the sons. The sons get the Y chromosome from dad. That's all dad contributes to them. So the trait they show for the X-linked characteristics depends solely on what the mother gives. Half the time, the mother's going to give her good dominant X chromosome. Half the time, the mother's going to give her recessive mutant X chromosome. So half the sons will have normal vision, and half the sons will be colorblind. Y-linked characteristics. There really isn't much to say about the Y chromosome. It's puny, it's sad, doesn't really carry much above and beyond the SRY gene. But the Y chromosome does some other things for us that are genetically useful, kind of straying a little bit outside of transmission genetics, Mendelian genetics. The Y chromosome is the most important thing on the SRY gene is the most important thing on the Y chromosome. As the SRY gene became so important for the species, crossing over, recombination during meiosis became more and more suppressed, became less and less prevalent, starting thousands, if not millions of years ago. This was to avoid gender switching. If you could get recombination and you got the SRY gene onto a different chromosome, you'd be transferring maleness to that chromosome too, and that's not good for many different reasons. So the Y chromosome kept shedding genes and getting different and different until finally there was no more recombination between the X chromosome and the Y. As the Y chromosome shed its genes and it got smaller and smaller, because it had less genes, it did less for the organism, for us. And so the genetic importance of the Y chromosome diminished over time as well. Only about a third of the entire Y chromosome contains genes, and the Y chromosome is very, very small to boot. Most chromosomes, the autosomal chromosomes, the X chromosome as well, encodes thousands of genes, many, many, many genes encoding many, many, many proteins. There's roughly about 175 genes on the Y chromosome, encoding about 175 proteins, and incidentally, many, if not the vast majority of those proteins, are for testes development. SRY kicks off the party of testes development, but many other proteins are needed to actually build those testes, and those proteins are encoded on the Y chromosome as well. So the Y chromosome makes testes, and that's pretty much all it seems to do. Other proteins seem to suppress some of the female traits that are promoted by the X chromosome. That's a little side note, but still promoting the development of maleness. So, at this point, there's really no homologous chromosome to the Y, and that means during meiosis, the Y chromosome has no one to cross over with. That raises a pretty interesting and significant genetic issue. Without any Y sister chromatids, without any synapsis between Y chromosomes during meiosis, the Y chromosome never becomes shuffled. Remember, that was the main point of crossing over during meiosis, was to shuffle these chromosomes up, throw all those genes kind of in the air and reassemble them into new chromosomes. That doesn't happen for the Y chromosome. So there's a constancy to the Y chromosome that no other chromosome has. The Y chromosome remains constant from generation to generation. Combine that constancy, that stability over time, with the fact that the Y chromosome is only passed among a single gender, passed from father to son, male to male, male to male, and what you've got yourself is a genetically based, what's called paternal lineage marker. You've got yourself a stable and genetically based DNA lineage passed only on the male side of a family. To put this into kind of everyday speak, the Y chromosome I have in each of my cells is nearly identical, almost certainly 99.99% identical to my father's Y chromosome. He gave it to me. But where did my father get that from? Well, he got it from his father, my grandfather. So his is nearly identical to his father's, which is nearly identical to mine. Where did my grandfather get his Y chromosome from? His father. And where did that man get it from? His father. So hundreds, thousands of years ago, there was a manila male who passed the Y chromosome on to his son, who passed it on to his son, who passed it on to his son, who passed it on to his son, until finally it wound up with me. The Y chromosome I hold in my cells is quite literally, nearly identical to the Y chromosome my male ancestor had thousands of years ago. For reasons we won't get into right now, mitochondrial DNA, the DNA that we find within mitochondria, acts very, very similar way as a maternal lineage marker. It has the same constancy and the same passage of generation, but only on the female line, from mother to daughter, mother to daughter. All of the autosomes are different. Each of us is a blend, is a shuffled mix of the chromosomes that our mother and father gave us on all of the autosomes. But when it comes to the Y chromosome, that was handed only from male to male, male to male. And then the same could be true of mitochondrial DNA for females. So, by sequencing the Y chromosome, and looking at unique sequences that tend to be different between different individuals, we actually have this way of creating what are commonly referred to as DNA fingerprints, or more accurately referred to as markers, DNA markers. We can track the lineage of mega male families, of large-ranging male families, track their migration across the entire planet, from Africa to Europe, then here to the Americas. We can identify living descendants of long-dead historical figures, such as the last Russian Tsar or Genghis Khan. We can even prove that Thomas Jefferson was a lying, cheating bastard, because there are African Americans present now that have Thomas Jefferson's Y chromosome in them. And the reason they have Thomas Jefferson's Y chromosome is because Thomas Jefferson contributed that Y chromosome by having sex with one of his slaves, and that slave giving birth to Thomas Jefferson's son. But of course that couldn't be known at the time, so that child was raised as a family of those slaves, and then continued on in the African American lineage in this country. And so we can track Thomas Jefferson's living African American descendants in this country now by using nothing more than Y chromosome technology. So you can think of the Y chromosome in this sense as almost a barcode. You look at a barcode, it has no information. You can get a computer to decipher a barcode, and it's just numbers. It's a code. It doesn't tell you the item or the price. It's a computer that makes that link. And then the Y chromosome's the same way. It doesn't contain information. It doesn't have a lot of genes. And even if we were to sequence a Y chromosome, it doesn't come out and scream at us and say, this is Thomas Jefferson's Y chromosome. We have to do comparisons. We have to make the linkages ourselves. We have to take Y chromosomes from African Americans who purport to be Thomas Jefferson's descendants and compare them to the Y chromosome, and this is how the study was done, compare them to the Y chromosomes of white Americans who know they are Thomas Jefferson's descendants, who can track their ancestry to Thomas Jefferson's family, and we see that those Y chromosomes, the codes in those Y chromosomes are identical, meaning those African Americans have the same exact Y chromosome from the same exact source as these living white descendants of Thomas Jefferson's. Just truly fascinating, unbelievable application of this type of information. And so all these Y chromosome markers are unique to the male lineage. They're not genes, they don't encode proteins, they're just sequences of DNA. They are codes containing this comparable information. Now this gets to something else as well. The value of Y chromosome analysis when we do human identity testing. Now we're truly in the forensic application of DNA. I won't spend too much time on this because I know not all of you are forensics majors, but still, this is an important application to this technology. The Y chromosome offers a striking and all-important benefit to forensic analysis. It's uniquely male. That's what we've spent much of this lecture saying. If you have a Y chromosome, you're male. And if you're male, you have a Y chromosome. And if you're female, you don't have any Y chromosomes. Most forensic cases are not murders. Most forensic DNA cases are rape cases. When an individual gets raped, they can submit a rape kit, and that's DNA evidence from the perpetrator, helping police to find out who has done this horrible crime. And the vast majority of rapists are males. I don't think that's a surprise to anyone. When we do get DNA samples from these types of cases, those samples are referred to as mixed. There is cellular debris in those samples that are from the victim, from the female, as well as cellular debris from the perpetrator, who was male. And so we have a mix of DNA in there, and forensically speaking, it's often very, very difficult to tell those two individuals apart when their DNA is mixed in that sample. However, when you have markers that are uniquely male, it's easy to identify and pick out the female markers from the male markers. And that's what we want. As forensic analysts and as law enforcement officers, we want the male markers. Those definitively belong to the perpetrator. So this is a pretty basic image of what one might see in a mixed sample. I won't get into the technology. You'll cover that certainly in your forensics courses of what these technologies mean. But just to give you some context, the simplest way to describe this is what you're looking at here is peaks from a genetic analysis. And you can think of each of these peaks representing either a chromosome or an allele. So here we have a criminal sample from a crime scene that has 3 peaks, 1, 2, 3, signifying 3 chromosomes or 3 alleles. 1, 2, 3. Now, obviously, there's no human being out there that has 3 chromosomes or 3 alleles. Or there's some rare exception to that, but it is very rare. And so we're looking at a mixed sample. We're looking at some DNA from female and some DNA from male. But we can't tell which is which here. Is the female homozygotic, and this is her peak in the middle, and the male is heterozygotic with 2 different peaks? Is the female heterozygotic and these 2 peaks belong to her, and the male is homozygotic and this peak is his one only type of allele? You can't tease out what's going on here. In fact, I'll show you what's going on here. If you were to get behind the scenes and somehow be able to pick it apart, which you can't do in the real world, but theoretically, if you could, with some kind of magic tweezers, pull the female DNA away from the male, you would see that the male is heterozygotic, and he has 2 peaks in these 2 positions. The female's also heterozygotic. She has 2 peaks as well. But this peak from the male is actually being hidden below the peak for the female, because the female DNA is that much more prevalent in the sample. So it gets difficult. This is for autosomal analysis. But now we do the same type of analysis and we look at the Y chromosome. And this is the profile, this is the information we get when we look at the Y DNA information in the sample. We get this. So we see it's a single peak in a particular location. Well, because it's the Y chromosome, we know that what we see here in the sample from the crime scene belongs to the perpetrator, because it came from the Y chromosome, and only the perpetrator had the Y chromosome. The victim, who was female, has no Y chromosome DNA. And so it allows us to identify DNA signatures, DNA fingerprints, that are uniquely and exclusively male. Very, very powerful in forensic applications. All right, so we're going to wrap up now with a conversation on dosage compensation. There's an added wrinkle in all of this that we can't ignore. Balance is important for every living thing. We have to all, and when I say we, I mean both gender types, we have to all exist pretty much the same way. We can't have too many genes, we can't have too few genes, and our cells need to function nearly identically. But you all have 2 X chromosomes, while I have 1. So again, a paradox. We have to be as genetically similar for our overall species health as possible, but here you guys have twice as much of a particular chromosome as I do. So how do we reconcile that? There's no balance there. You have 2 and I have 1. How do you make a single species survive in almost an identical genetic and physiological way when one gender has twice as many genes for an entire chromosome set as another? And that's not necessarily a rhetorical answer. That's a real question I'd like you to get at. What do you think is the fix for this? Well, the problem is referred to as dosage compensation. Dealing with this problem is a dosage compensation solution. What we mean is, if we've got a difference in dosage, females have 2 X chromosomes and males have 1, then we need to compensate for that difference in dosage, and different species deal with this differently. We'll talk about only humans and mammals, simply because we're running out of time, and that's what's most important to us anyway. No matter how it's done, the way I'm about to tell you for humans and mammals or other ways, dosage compensation, the end result of dosage compensation, is that it equalizes the amount of protein made from X chromosome genes. So even if males have half as many X chromosome genes as females, dosage compensation fixes that by getting the same amount of protein made in male and female cells. And remember from that very, very, very first lecture, seemingly a while ago, it's all about the proteins. If you make the same number of proteins, you have the same cellular effect. So in 1954, a male scientist, a man named Murray Barr, identified these dense things. They appeared to be very dense, very heavy. They stained very easily with DNA stains. And he observed them in the nuclei of female cat cells, not in the cells of males. He didn't know what they were, but he was somewhat narcissistic, and so he named these structures after himself. They were referred to and are still referred to as Barr bodies, but that's all that he did. He didn't characterize them. He didn't say what they were. 12 years later, in 1961, a much more successful, much more preeminent and capable scientist named Mary Lyon finally actually figured out what the hell these Barr bodies were. Unfairly, they're still referred to Barr bodies, even though Mary Lyon is who characterized them, although what I'm about to tell you is referred to as the Lyon hypothesis, named after her. She was extremely insightful in what she came up with for this, and what she thought was true wound up being proven true not long after. In general, women scientists have contributed enormously to the life sciences. For STEM fields in general, I'm sure you're aware that females have often had a hard time breaking into and being accepted in these male-dominated fields, unfairly so, of course. There are plenty of horror stories in biology as well, some of them quite famous, but in the general defense of the life scientists, females have had a much, much easier time contributing and being successful in the life sciences as any other STEM field, and I think it's fair to say right now in the life sciences there's almost complete integration between the genders in terms of the contributions that scientists can make to the field. But back to Mary Lyon. So Mary Lyon hypothesized that these Barr bodies were actually very densely compacted, completely inactivated X chromosomes. And the reason why that they were in female cells only is because females have 2 X chromosomes, and males only have 1, and Mary Lyon was absolutely correct. In every mammalian female cell, there are 2 X chromosomes present. In every mammalian male cell, there is 1 X chromosome present. That is a dosage problem, and it needs to be compensated for. The way that mammalian cells compensate for this is that in female cells, 1 of those 2 X chromosomes, and which one in any given cell is always random, 1 of those 2 X chromosomes is completely deactivated. It's taken completely out of the equation by being wrapped up so tightly and so densely that the DNA is inaccessible. So while all of you guys out there are technically diploid at your X chromosomes and at your X genes, technically you have 2 copies of each allele, you are really all functional haploids just like me, because 1 of those chromosomes has been completely inactivated in each of your cells. So you say, well then, the whole color blindness thing, what the hell was that about? You told me that females have much less color blindness because they could be heterozygotic. Well, which cell has it inactivated is always random. So if you are, for argument's sake, a carrier for color blindness, that means you have one dominant allele on one X chromosome and one recessive allele. In your eye, half of your retinal cells have deactivated the mutant X chromosome, leaving the wild-type allele expressed, and those retinal cells can sense color. And half of your retinal cells have deactivated the wild-type dominant allele X chromosome, leaving only the mutant colorblind allele expressed, and those retinal cells are technically colorblind. But because you have a mix and match of both, you can sense color. Maybe not as well as a normal visioned person, but you can see the differences in colors because half of your retinal cells are expressing that wild-type normal allele. So sure, one of your eye cells is colorblind, but the cell right next to it inactivated the other X chromosome, so it's expressing the normal proteins, and so in general, you average out to the dominant phenotype. The normal cell, with the normal X chromosome active, helps out, picks up the slack for the other cell, which has the normal X chromosome inactivated. But sometimes, we can see really complicated genetics with our own eyes. The gene that's responsible for orange tabby, orange fur color in cats is X-linked. So orange tabby cats have that orange color because of an X-linked trait. Male cats only have one X chromosome, and it's the same exact X chromosome in every single one of their cells. So male cats are either orange tabby or they're not. But female cats have two X chromosomes. And in those cells of those female cats, if one of those X chromosomes encodes orange tabby and the other doesn't, in each individual cell, one of those X chromosomes is inactivated and the other isn't to achieve dosage compensation. So what you get in these cats is a fur pattern that we sometimes refer to as calico, sometimes we refer to it as tortoiseshell. We get this. I take one look at this beautiful animal and I know she's female, because she's expressing orange tabby fur on some of her cells and not others. Here is orange tabby coloration here. If this was a male cat, the only information that this male cat could possibly have if he was showing orange tabby would be orange tabby. And so the whole cat would be this pattern. This cat seems to have a cellular choice. Orange tabby in some patches and not orange tabby in others. By seeing that, I know this cat has two X chromosomes. In the orange tabby areas, one X chromosome was inactivated. In the non-orange tabby areas, the other X chromosome was inactivated. Two X chromosomes means female cat. Any cat with this kind of mixed orange tabby and not orange tabby coloration must be female. Must be female, because we are witnessing the effects of X inactivation with our own eyes. Sometimes each orange patch can be a small patch. Sometimes the orange patches are quite large. Sometimes we see half the cat orange tabby and half the cat not. It's quite rare, but it can happen. The reason for this is because each orange patch represents a population of cells that arose or were born by, were the mitotic result of a single X inactivation event in the non-orange tabby gene, and so there's a clonal population or a daughter population of cells that all have the non-orange tabby allele inactivated. Other cells have the orange tabby allele activated, and they give rise to a clonal population of cells that all express orange tabby. Inactivation occurs fairly early on in development, and so we don't have X inactivation occurring later in development. These cells don't switch from one to the other. Once you establish that pattern of X inactivation, that pattern remains inherited for all the daughter cells that come from that lineage. This cat, this is touching this year for me. When I was first reviewing these slides, I forgot this picture was even here. This is Frankie. He was my cat. This picture was taken last year during spring, right around this time of the fall semester last year, and unfortunately Frankie died this winter, so here he is, and in all his glory. He was a psychotic cat, very, very violent. We couldn't keep him inside because he was just, he had significant emotional and psychological problems, but he was a hell of a good cat. And I show his picture here because we can take one look at Frankie and see that he's male, regardless of his name or if we know anything about him. He's an orange tabby cat. His entire fur coloration pattern is orange tabby. The reason why Frankie is showing nothing but orange tabby is because he only had one X chromosome to work with. No X inactivation, no dosage compensation. He only had one X chromosome. The trait on that X chromosome was expressed in every single one of his cells, giving him a consistent and thorough orange tabby appearance. How can you not love this stuff? I mean, how can you not at this moment be sitting there and saying, well, I don't get it all. Some of that was boring, some of that was dry, some of that went over my head. But you're telling me I can look at some cats and know their gender from their color pattern? I think that's pretty friggin' awesome. So what do we talk about in this lecture? Started off very basic. We have this chromosomal gender determining system. In us, in humans, and all mammals, the 23rd pair of chromosomes are the sex chromosomes. They determine the gender. Males are defined as males because they make smaller gametes. Females make larger gametes. Most sex determination is chromosomally based, is governed by these chromosomal sex determining systems. Sex is genetically determined. In humans, the presence of the Y chromosome makes you male. If you have a Y chromosome, you're male. If you don't have a Y chromosome, you're not. And this is because the Y chromosome carries the SRY gene. The SRY gene sees to the development of testes. Those testes make testosterone very early on in development in utero, and that testosterone makes you male. The default state of humanity, left unchanged and unchallenged by hormones, is female. And any developing human, regardless of their genetic composition or the presence of their Y chromosome, if their cells can't sense the presence of testosterone, they go down that default route to female. The implications of all this is that males are haploid for all genes on the X chromosome because the other half of that pair is Y, and the Y chromosome really doesn't contain much. So males have one allele and one allele only for all X chromosome traits. Genes on the X chromosome are referred to as X-linked because they are present on the X chromosome, and we introduced X-linked traits using the white-eyed fly example that Thomas Hunt Morgan first found. For human beings, we use the example of red-green color blindness. The gene that allows humans to see red-green spectrum light normally is found on the X chromosome, making color blindness an X-linked trait. Females have two alleles for this trait, two shots for this trait. They're diploid for this trait, just as all humans are for other traits. But males only have one X chromosome, so males only have one allele for color blindness. Males only have one shot to get this allele right. The best and simplest way to say that is males express the trait of their one allele. We talked about the Y chromosome as a paternal lineage marker. We can track the lineage, the male inheritance pattern, for generations and generations ago all the way to living males now, because the Y chromosome is passed only from fathers to son, and with no other Y chromosome to recombine with, the Y chromosome is very constant. Having these markers that are uniquely male not only allows us to do these lineage studies for generational identifications, like I said, Russian Tsars and Thomas Jefferson, but also has forensic implications where we can identify uniquely male DNA even when it's mixed and contaminated with female DNA from a crime scene. We ended the lecture with this idea of dosage compensation. In mammals, we call that X inactivation. An inactivated, tightly wrapped X chromosome is a bar body, but it's Mary Lyon who actually figured out what these bar bodies were. The purpose of X inactivation is to reduce the functional X chromosomes in females in half, so that females have one functional X chromosome in each of their cells, just like males do. But because this X inactivation occurs randomly, females are a chimera. They're a mix of cells that contain one active X chromosome or the other. So a lot of these X-linked traits don't manifest in females because there's usually another cell nearby that can do whatever the mutant cell couldn't do. So that's it for sex-linked traits. We're going to move on to Chapter 5. We actually split Chapter 5 in half. So the next lecture will cover the first half of that chapter, a continuation of the conversation of changes or perceived changes to Mendelian genetic rules. After the end of the next lecture, we will pause for the exam, and then we finish Chapter 5 up, Part 2 of this lecture series, as the beginning of Exam 2 material. So this chapter that's coming up, Chapter 5, is actually cut in half with an exam in the middle, but we'll talk about that a little bit more as we introduce the next lecture. As always, thanks for watching.